Hello everyone. Welcome you all to my YouTube channel Let's Do Biology. I hope you all are doing well. If you are new to my channel, then please subscribe my YouTube channel Let's Do Biology. And if you are liking my videos, then hit the like button and don't forget to share it with your friends. So guys, again welcome to my channel. So in this video, I will discuss my PhD interview experience at IISc of Biochemistry department. In previous video, I already discussed my interview experience of MCB and DBT department. In this video, I will discuss my biochemistry department interview of first round and second round, in which I got selected. Okay. So let's get started now so that you will get some experience from my interview experience and get some idea that how you need to prepare and what you need to prepare and in which way you can answer your question, in which way you can frame your question. So for biochemistry first round, these were question which were asked by professor. So in my panel there were like four professors were there for my first round of interview in my panel. So let's discuss these question. सेम लाइक अर्लियर, ये भी मैंने जैसे मेरा इंटरव्यू हुआ आई जस्ट रोट ऑल द क्वेश्चन विच आई रिमेम्बर एट दैट पॉइंट ऑफ टाइम, so that it is now very easy for me, so that I can give you idea about the question. Okay, so uh, uh, there were three professors which asked me question, and among them one professor which didn't ask me. Okay, so let's get started now. In first round of interview, for one professor asked me ki what are your papers in masters, and then they were like, she asked me all of the questions like from my papers, my master's paper, as I was having genomics in my final year of masters. So she asked me, then one of my papers related to gut microbiome, then she asked me what have you studied in microbiome. Okay, then I generally told about the overview or like broad topic of my paper, gut microbiome. Then she asked me, how will you determine different microbiome, like tell me which technique you can use for determining that which are the microbiome are present in your sample. Okay, so then I explained one or two techniques, but she was asking, but the professor want to ask me more technique, but I was not able at that time to tell her more technique. Okay, so I just explained like something two or three techniques, uh, maybe two technique. Okay, then further she asked me, tell me some difference about genome and metagenome. What is the basic difference that you, by which you can define genome, or like what is metagenome? Then I give the general definition of these genome and metagenome. Then further she asked me, if you have a mix of substance or like media substrate, if you have media, mixed media, then how will you determine about each bacteria? Means आपके पास एक मीडिया है जिसमें बहुत तरह के वायरस है, या फिर मतलब लाइक कोई भी मीडिया है, बैक्टीरिया है, डिफरेंट टाइप के, तो आप हर एक बैक्टीरिया के बारे में कैसे पता लगाओगे इफ यू डोंट हैव एनी इंफॉर्मेशन अबाउट इट. ओके, then I told her that we can use in silico approach and give some more information. I haven't remembered now what I told her. Then further she asked me, uh, because one of my subject in masters was epigenetics in final semester. I was having genomics. There were four paper in genomics. So one was gut microbiome, and the next one is epigenetics. So she further asked me about epigenetics, that what is epigenetics. Like, I think the other professor asked me question about epigenetics. He asked me what is epigenetics and what are the different type of modification which comes under epigenetics. Okay, then he asked me what will they do. Okay, like what these modification have their function, like what are if the our structure of DNA or like protein is modified, then what is their function, what will they do after modification. Okay, and like the different enzyme which catalyze this modification, can you tell the name of some enzyme? So I told him different type of enzyme like hat, demethylase, methylase, phosphotransferase, and glycol. And then I also told him like these modification: acetylation, methylation, phosphorylation, and all ribosylation. Okay, then further, the same professor asked me how methylation occur. Like, what is the way that how is this occurring methylation, and tell me the enzyme which carrying out this methylation. Okay, like it is methylase enzyme, and if it occurs on DNA, then it is DNMT, like DNA methylase enzyme, DNA methyl transferase. Okay, and this methylation, how it affecting the gene activity. You all know ki if methylation will occur on our DNA, then it will suppress the active gene activity, the expression of the gene. Okay, so I just told this कि how it affecting the gene activity. And then further he asked me which protein will bind. If methylation is occurring on DNA, then further which protein will bind to that methylation part. So then I told him ki it is like HP1, like heterochromatin protein 1. Then he told me then that is there any, you know about MBD protein? Then I said yes sir, methylase binding protein. Yes, methyl binding protein, it is also the protein that bind to that methylation part. Okay, or like MECP also. Then further what he asked what is the function of DNA methylation in which enzyme. So further he the same thing I have written here ki which enzyme is carrying out and how it will cause condensation. Okay, he asked me about the like in which way it is carrying out the condensation, because I just told no कि it is suppressing the gene expression. So how it is suppressing by causing condensation of the chromosome, ठीक है, chromatin. Then he asked me, do you know about CPG? Then I told him about the CPG island. And also that professor asked me how much amount of like CPG consist how much constitute how much amount of our chromosome. This type of question I am actually not correctly remember at this point. Like he told me around 60 to 7. I told him ki 40%. Then he told me ki percentage of CPG 60 to around 70 or like 80%. ठीक है. And then he asked why it will not cause methylation in promoter. Okay, okay. Then he asked if CPG because methylation occur on this ना, तो he asked why it will not cause methylation in promoter. promoter में क्यों नहीं होता, क्योंकि promoter ही हमारा ट्रांसक्रिप्शन कराता है ना. अगर उसी में कंडेंसेशन हो गया तो ट्रांसक्रिप्शन प्रोसेस होगा ही नहीं. So that's why it is not occurring in promoter region. Okay, then these question like from these were asked by one professor and the other from here, the other one, and finally these were asked by one more. Okay, then what he asked, like I think Mahipal Ganji, Dr. Mahipal Ganji asked me this, that what type of interaction occur between DNA and protein. Okay, so I told him hydrogen bond. Then I told him electrostatic also. Then he was expecting electrostatic from me. Then I explained how these electrostatic bond form in DNA and protein. Like I explained about the charge, about charge. Okay, phosphate have negative charge na, and protein like positive charge. So it will form like electrostatic interaction. Then further he asked me how can we determine the affinity of okay, interaction affinity हम कैसे determine कर सकते हैं, but I was like confused at this point. Then he asked me difference between nanomolar and micromolar, like he was expecting it is related to this question. Exactly I am not able to remember it, but it was something related to the earlier question. Then he asked me among nanomolar and micromolar, if I ask which has high affinity, then which one is this? Okay. Then he asked me, do you know about KD and Ka, like dissociation or affinity constant? So I explained this, and last he asked me if do you know if there any equation related to this dissociation or association constant, but I was not able to remember at that point, so I haven't answered this question. Not. Okay, so this was all about my first interview. Then I got selected in first interview, and for biochemistry in second round of interview, these were the questions. Okay, so like in second round of interview, they were around 8 to 9, like 4, 5, 6, like yeah, 7 to 8 professors were there in my panel for second round of interview. Okay, so it was like two professors had not I think yeah, two professors didn't ask me any questions. The rest of the professors asked me. So the questions were like, first they like in all of the interview, in all of this department interview, first they will ask your topic of interest. Then later they will ask you a question, and that can deviate from your topic of interest. Like in my first round of interview biochemistry, all of the questions were like from my master subject. They were not, they don't even ask me my topic of interest. But in second round of interview they asked me. Then I told ki cell biology and immunology. Like in all of the interview I was like my favorite topic of interest is cell biology and immunology. And in some interview I got question from this topic, but in other I got the question from like different topic also. But so you have to prepare for all these type of condition. You can by luck you can ask from your interest of topic, or otherwise you can ask from different topic also. So you have to prepare this way. So so let's see, for the second round of interview the first question was how to generate. So this was from immunology, and मैंने वैक्सीन वैसे तैयार प्रिपेयर इतना अच्छे से लाइक नहीं किया था, बट स्टिल दे आस्क मी फ्रॉम वैक्सीन. So the first question, how to generate hepatitis B vaccine? Like what is the strategy to generate hepatitis B vaccine? So I said कि recombinant technology. ठीक है. Then further they asked me about the procedure, how you will generate or make hepatitis B vaccine. Okay, so I said ki by using some vector or something like that. I gave the answer like this. So then they asked me about like vector, can you please give the name of some vectors. So then they were like कि वो रिकॉम्बिनेंट डीएनए टेक्नोलॉजी में घुस गए. ठीक है. सो they asked me, do you know about PBR 322? So I said yes, and like some more vectors. So let's go and leave it. The next question is, do you know about corona vaccine? Can you please tell us the name of two vaccine which were given, generally given to the people, and which was given to you? So I was like, literally I was blank at that time, that what should I give the answer. Then further, but luckily मेरे को याद आ गया था, so I gave the answer of this. Then they asked ki these like covaxin, I gave na answer covaxin. So they asked me ki which type of these vaccine. Okay, and how this vaccine were generated. So जब भी किसी भी इंटरव्यू में जाओ, तो अभी जैसे कोरोना हुआ था, तो इसके बारे में अच्छे से पढ़ लेना इंटरव्यू के लिए, तो बहुत ही अच्छे से कि कौन सा वैक्सीन था, उसमें कौन सा क्या चीज यूज किया गया था, कौन सी स्ट्रेटजी यूज की गई थी. ठीक है. तो ये बहुत अच्छे से याद कर लेना, पढ़ लेना इस टॉपिक को, ज्यादा टाइम नहीं लगेगा, 2-4 पॉइंट याद करने की जरूरत होगी, और उससे दैट्स ऑल आंसर दे पाओगे आप. Then what he asked the professor, how body responds to a live antigen. Okay, okay. Then the other professor deviate to like MHC from immunology, that how live antigen in our body, how a body responds to a live antigen. ठीक है. So you all know ना कि MHC1 give response to virus, that live antigen. So I gave the answer of that MHC1. Then they further asked me name some APCs. So I gave the name of three APC like B cell, macrophages, dendritic cells. So they further asked me, is there any other APC? So I said there are some non-professional APCs also. So I also gave the name of some non-APCs also. Sorry, non-professional APCs. These three are professional APC. Then further, the other professor asked me, like I told ki cell biology one of my topic of interest. Then he asked me the question from cell biology, that cell wall, do you know cell wall is the cell wall constituent, so cell wall is made up of. Then I gave the answer, and I then he asked me which type of protein present in cell membrane. So first first of all I confused ki in which context he asked me ki which type of protein. Then but luckily I got ki peripheral or integral protein. Then then I said ki he said ki make a diagram. Peripheral protein are present only on the outer side. He asked me this question, and he also told me that make the diagram of the cell wall, roughly made the diagram of cell wall, that how is how their constituents arranged like. Okay, then I made it, and label all the like peripheral protein, integral protein, phospholipid and all. Okay, then further, yeah, he said label cytoplasm and extracellular side, both cytoplasmic side and extracellular side. Or ask, I like I label peripheral protein on the outer side. So he asked me like the, are these peripheral protein only present on the extracellular side, or like present on the inner side also? So I said yes sir, these are present on both sides. So he said yes, why the, yes, but one professor confused by my answer. Then, but like Professor Utpal Tatu was there in my panel, so he gave the idea like what I am telling, what I am trying to say. Okay, then further, what was the question? How can we isolate? Then the other professor asked me, how can we isolate peripheral and integral protein? Then I gave the answer of this question also, like different for peripheral and integral also. Then I gave ki we can use detergent for integral protein. Then he asked, is there any other method by which you can isolate them? But I was not having the idea of that. Then he said, I also don't know, you I just want to that if you also know or not. Okay, so okay, so मेरे को भी नहीं पता था, and he was like, can you tell me? I was like, no sir, I don't know. Then further, how can we determine that our protein is targeted in ER? ठीक है. Then I also here I give some technique and explain those. Then how protein folding occur? Protein folding कैसे होता है? Okay, I was able to answer this question also. Then further, do you know about calnexin and calreticulin? Which one asked me? I actually forgot the name of the professor that which one asked me this question. Like, I think it was Professor Patrick De Silva. He asked me, "Do you know about calnexin and calreticulin?" And I was pretty enough to give the answer of this question also. How they help to, अच्छा, like फिर उन्होंने पूछा कि कैलनेक्सिन और कैलरेटिक्यूलिन है कि ये कैसे फोल्डिंग में ये कैसे हेल्प करता है, कैसे डिटरमाइन कर सकते हैं हम इनसे कि हमारा फोल्डिंग हो गया है नहीं. फिर मैंने वो भी पूरा प्रोसीजर एक्सप्लेन किया, एंड it was like also correct. And then further he asked me which different modification occur on protein. Okay, like he was expecting this like N-acetyl galactosamine, glutamate, sorry, glucose or mannose like. Okay, then further, how many मैनोज present after trimming? Trimming के बाद कितना मैनोज बच जाता है जब like modification होता है ER and all गोल्गी में. Then उसके बाद, then further give different enzyme and their role. अच्छा, then सर ने क्या किया, मेरे को बहुत सारे एंजाइम बताए और पूछा कि इनका रोल आपको पता है. बट आई वाज लाइक, कुछ मेरे को पता था, सारे नहीं पता था, बट विच एवर आई नो आई गेव द आंसर ऑफ दैट ओनली. Okay, you don't need to give answer of all the question. They just want to know कि like आप कितना आंसर दे पा रहे हैं और कितना अपनी तरफ से ट्राई कर रहे हैं. Okay, so don't be panic about like you don't give the answer of all the question and your viva, sorry, your interview was not that much good like you want. It was same with me, but okay. Then further, which enzyme? Okay. Then he asked me, like ये modification जब होता है, trimming होती है, तो ग्लूकोज जो होता है उसको कौन सा एंजाइम रिमूव कर सकते हैं? Okay, glucosidase. Okay, so guys, this was this much only for my interview in Department of Biochemistry at IISc. Okay, so अकॉर्डिंगली जैसे क्वेश्चन आपको पता चल रहा है इसमें से, उसके अकॉर्डिंग आप अपना प्रिपेयर कर सकते हो, और जो भी आपका इंटरेस्ट ऑफ टॉपिक है उसमें से उसको अच्छे से प्रिपेयर कर लेना. ठीक है. ऐसा नहीं होना चाहिए कि लाइक आपको कुछ पता हो और कुछ नहीं पता हो. जितना पढ़ो उसमें से उसको अच्छे से तैयार कर लेना. पढ़ना तो सारा ही है, लेकिन जो जो यूनिट जो चैप्टर एक करो या फिर दो करो. ऐसा नहीं है कि आपको दो ही बताना है या फिर तीन ही बताना है. यू कैन डू योर अकॉर्डिंगली योर इंटरेस्ट, कि आप एक बताना चाहते हैं या फिर दो बताना चाहते हैं या फिर तीन, वट एवर यू वॉन्ट. बट अगर एक बताओगे तो उसमें से ज्यादा डीप में से जाएगा और उसको आपको अच्छे से प्रिपेयर करना पड़ेगा. ठीक है. तो थैंक यू so much guys for watching my video so calmly and patiently. So guys, please subscribe my channel Let's Do Biology for getting this type of video, so that you can get benefit from all this type of video. Please like, share and subscribe my channel Let's Do Biology. I hope you are liking my video. We'll meet you in next video. Till then, take care. Bye bye.